During our discussion of glycolysis, we examined the energy landscape of the process, and we noticed 3 steps where the process is irreversible. The first one being the conversion of glucose to glucose 6-phosphate. And we talked about how that's important and relevant because this is a great place for branching off of this metabolic pathway. And that's exactly what we observe when we think about glycogen synthesis. The process of making glycogen starts at glucose 6-phosphate. In this video, we're going to go ahead and examine the steps required to convert glucose 6-phosphate into glycogen. To start this off, I first want to note that I'm going to start taking shortcuts when I draw these phosphorylated sugars. Okay, I'm no longer going to be drawing out the full phosphate group as I show here in glucose 6-phosphate. Instead, I'm going to be abbreviating the phosphate group with a capital P that's circled. Okay, so as we move through the rest of this class, just note that if you see a capital P that's circled, that means a phosphate group, PO4. All right, so with that in mind, let's go ahead and think about how glycogen is synthesized. This process starts off with glucose 6-phosphate, which is the second molecule found in the process of glycolysis. Glucose 6-phosphate is converted into glucose 1-phosphate in an isomerization reaction using the enzyme phosphoglucomutase. So the only thing that's happening here is the phosphate group on the 6th carbon is being transferred to the 1st carbon. We go from glucose 6-phosphate to glucose 1-phosphate. The second step of this is known as an activation step. So what we are doing is taking our glucose 1-phosphate and activating it to be able to participate in the next reaction. Okay, we do this by reacting it with uridine triphosphate, or UTP. So UTP is really similar to ATP, except in this case, instead of adenine as the base group, we have uridine. In this reaction, we are transferring uridine monophosphate, or UMP, onto our glucose 1-phosphate. And we end up with an intermediate that contains 2 phosphate groups linking together the uridine and the glucose. This molecule is known as UDP glucose. The final step of this process is linking this activated glucose onto a growing glycogen molecule. As we briefly mentioned in the past, glycogen is a long polymer of sugars linked together through glycosidic bonds. And specifically, it's alpha D-glucose units linked together through a glycosidic bond between carbons 1 and 4. And the idea in this image is that the R I'm showing represents a much longer chain of glucose molecules all linked together through this same bond. Notice that we do have one free alcohol group present on carbon number 4 on the terminal glucose sugar. It's this alcohol group that can initiate the next glycosidic bond with our incoming activated glucose. When this reaction completes, we end up with 2 products. One is a glycogen chain with an extra glucose molecule that came from the UDP glucose. The other product is uridine diphosphate, or UDP. The enzyme that carries out this reaction is known as glycogen synthase. And I want you to keep this enzyme in mind because it plays a really important role in how glucagon and insulin are able to regulate glycogen synthesis and glycogen breakdown.